I will try to get you up to scratch with this area of quantitative analysis of genotoxicity data for risk assessment purposes and tailor it to your area of interest as well. It can be interactive. There's a part in this talk where I'll show you how to do some dose response modeling. And if you don't generate data yourself and do dose response modeling or stats yourself, this should still be relevant because you'll still be analyzing data that someone else has published or that they've made decisions based on these sorts of approaches too. So really, it is an important thing to be able to understand and be able to critique and be able to point out when people have done this correctly or incorrectly. So I'll get straight into it. So I'm associate professor at Swansea University, and I've been working on this sort of topic for a while. I started out really thinking I had quite important dose response data, and people were saying you've got to get good at stats. Like David Kirkland was like, oh, you've got to get good at stats, or this is sort of meaningless. And that was about 20 years ago. So I've been over the years trying to get a bit better at this and trying to progress the whole field towards a more risk-based approach as well for in vivo data, and then for in vitro data, trying to get as much as we possibly can out of all of this data as well. And some of these approaches I'll be talking about really improve both the in vivo analyses and the in vitro analyses quite considerably. So as I said, my area has really been just to move away from the paradigm that all of these chemicals, all of these mutagenic carcinogens at any dose are going to kill us all off, because that's just nonsense. So I look at some previous work on this area, like Bruce Ames, who invented the Ames test, and Lois Gold, who used to have the Gold database. It's called the cancer potency carcinogenic database. I think it was called that in Berkeley, and that now resides in LHASA, LHASA. So that's where all the cancer data that Amaya was alluding to is actually kept, and Lois was one of the first authors of that. So these are pretty high-hitting people, and even back then, when they wrote this, quite a sort of jovial, quite a fun sort of indication. It was really a pamphlet, I think. They sort of try to highlight that all of these carcinogens that we're risk assessing, most of them we already intake from in a natural way. So man-made's a very loaded term, and natural is said to be super safe. And that's not really the case. We all know that in this room, but it's good to highlight that at the beginning of one of these risk assessment talks, that we're not trying to poison the whole population by giving them loads of carcinogens. We're trying to ensure that people aren't exposed to high levels of potent carcinogens, but really that's done through risk assessment, not just banning every single product known to man, because that's just completely mental. So it's really good. So things like butter, coffee, all those tasty things have got benzene in it. We know benzene's not too great. We got benzoate pyrene. That's all in all the tasty things as well. So coffee, as I said, it's got about 20 known carcinogens. So are we going to ban coffee? No, we're not. So let's just be sensible and be sensible. Use that thinking cap when we look at other mixtures of chemicals in the different products that the human population are exposed to. So that's the sort of introduction to why I'm so interested in ensuring that we're not exposed to high levels of everything. And that we do actually start understanding dose response and mechanism. With these groups of different genotoxic substances, the mechanism is very important. Under current guidance, there's a couple of groups of genotoxic compounds that are said to have thresholds. So the main one of these would be antigens. So those ones are the ones that interact with spindle microtubules during cell division. And multiple microtubules would need to be affected before the chromosome is lost into the cytoplasm, or some non-disjunction or something happens. So that'd be an antigenic event, a numerical event, and that's supposed to have a threshold where low levels of antigens are okay. And we know that from many drugs that we expose ourselves to and our kids to, such as mebendazole in threadworm tablets that you give to your kids when they've got threadworm, actually are just like 400 grams of an antigen, and that's been fine to date. So we sort of realize that this is true, and we can back this up with evidence. But antigens are one of the first ones, and there's a few other mechanisms as well. Some of the more recent ones that have been accepted by regulators are things like DNA synthesis inhibitors, substances that disturb nucleotide pools, direct acting alkylating agents, and PAHs, etc. But alkylating agents are the ones that are most loaded. They are often very problematic in the pharmaceutical industry. they pop up quite a lot. They're quite small. They just add an alkyl group onto your DNA, and they're quite small, so they can get into the major and minor helix of your DNA's curve like that. So you can get into even the really tightest nooks and interact with your DNA. So they are problematic, but there are ways that low levels can be withstood. So in our table, when we worked on another expert work group, this was Brazil in 2013. I think it was 2013. That was quite nice. So that was Iguazu Falls. You'll realize that our scientists, if you're early in your career, this is quite a big benefit to being a scientist in this area. You get to travel quite a lot, and we've seen quite a lot of nice places. I sometimes share a nice map of all the different nice places we've been to on these conferences. But this one was lovely. It was in Brazil, Iguazu Falls. And at that one, we had this different group of experts from people like David Eastman, worked for the EPA quite a lot. He's an academic. We had Food and Drug Administration people, European Medicines Agency, lots of different industrial people, and a few academics. We wanted to show that there were actually case studies where some chemicals have been shown to have thresholds. And these are the ones that we've got. So this is a nice publication to cite when you want to say, oh, there have been examples of even DNA reactive substances having accepted thresholds. I say threshold because I'll say why I don't really like that term in a minute. But threshold mechanism is something I can link to, because threshold mechanism is more at certain levels, DNA repair can withstand all of this, or at certain levels, the chromosome isn't lost. That's a more conceptual thing that you can talk about scientifically. A threshold dose where nothing happens and then something happens is almost impossible to define, and I'll show you that in a minute. But this is quite nice. It sort of alludes to this thing. All these chemicals aren't going to kill us at all these different doses. There are levels of nearly every single type of compound that can be withstood. You just need to prove how that mechanism comes about and at what doses those levels are that we can withstand. And it's quite tricky for some compounds. Some people never agree that DNA repair can withstand low levels of compounds, so it's usually quite argumentative. So it's been quite fun, my career to date. Following on from Amaya's talk, I put this one back in. I thought it was quite interesting and useful. She put forward all the different genotoxicity tests that we can use to assess compounds, lots of the in vitro ones, and then I'll just highlight the in vivo ones here. So when I'm talking about risk assessment based off genotoxicity data, to date, it's really just in vivo data that we would support. And at some point, the in vitro data would catch up, and you would be able to do risk assessment on those. But at the current time, it's a few years away, I'd say. So really just highlight what the in vivo tests are, mostly for this first step in proving to people that there are these low levels that can be withstood. Actually, it's been the micronucleus test for the antigens. For the micronucleus test, you can show the nonlinear dose response. You can even, some people have defined threshold doses, but I'll show why some of those calculations aren't too great in a bit. For some of the more pokey ones, the more important, more potent, more problematic ones, they would be causing G mutation. So we probably know that Ames positive compounds are a bit of a nightmare. If you're in the pharmaceutical industry, and Ames positive usually kills your drug. However, things like the quantitative structural activity relationships in QSAR have enabled companies to be able to just avoid Ames positives, because they just put their list of chemicals through their QSAR and it identifies what going to be positive or not. Some other fields, this isn't the case, and you do get quite a lot of Ames positives. So if you've got an Ames positive and you want to show that, say, humans aren't going to be exposed to dangerous levels of these, you could do dose response analysis in this in vivo transgenic gene mutation test. So these are animals that have had a transgene inserted on a plasmid. These animals are treated. That plasmid's then taken out afterwards, grown up in bacteria, and then you score the bacterial cells. So it's a bit strange. It's a bit hard. Not all labs can do it. But this is being activated quite a lot for the nitrosamine situation for the pharmaceutical industry. And that has a tail of probably supporting EFSA as well, who've got some recent guidance on nitrosamines, and ECHA as well, who've also been updating their nitrosamine guidance. So watch this space for transgenic gene mutations. ECNGS is hot on the heels. This is really probably what's going to overtake everything for me in genotoxicity testing. So it can be any gene, it can be the whole gene. At the current time, it's panel of 20 loci within the genome, cross-genic and non-genic. You can do dose response, you can get mechanism and everything. So when I'm talking about doing risk assessments on genotoxicants, genotoxic carcinogens, it would be from these in vivo data, and probably not these ones so much on the right. They would have different issues that we could go into, or this one, which is only just one tissue. So when we're thinking of this, and when you're thinking of analyzing your own data or analyzing something on someone else's publication, we put forward this as well in our Brazil IWGT meeting. So we wanted to see what the best way of defining this small increase above the background or no increase above the background was. We called it a point of departure. I think EFSA called it a reference dose. It's the dose at which sort of nothing's happening, or a dose at which you can say negligible effect or no extra risk. There's loads of different terms for it, but it'd be like a point of departure or a reference dose. So there's different ways of defining this. Benchmark dose, you can do this, and I'll talk about this quite a lot. No observed effect level. In our area, it's genotoxicity, so no observed genotoxic effect level. Sorry about that. And then this is the threshold model, the bilinear model. So we analyzed all of this and saw which is the best, which is the worst, what should we do in the future. Really came out the benchmark dose is the only one that should be used. These other ones are really problematic for point of departure use. We realized that for hazard characterization, for yes, no, people will still use pairwise testing. So benchmark dose is statistical modeling. I'll show you a lot in a minute. No observed effect level is just pairwise testing, like a t-test or a Dunnett's test. Yes, something's positive or not, and then you go through each doses and see which is the first significant dose. No observed effect level is the last non-significant dose. Bilinear is a threshold model. So that's what these things are. And we came out that the benchmark dose is far superior for many reasons that I'll show you in a minute. I think some of the major disadvantages. So this is quite a big one that we came out with. So pairwise testing, I'll show you again in a minute with a figure. Pairwise testing to find this sparse data, so not very good data with like massive error bars. That's what sparse data means. Tends to yield higher points of departure. So what this means is if there's two studies, one's rubbish. So for the same chemical, one's rubbish and got massive error bars, you'll get a higher no observed effect level for that. So if you do a risk assessment on rubbish data, you end up exposing the human population to higher levels of that chemical, which is absolutely awful. It means it's not protective. So the no observed effect level is not protective for risk assessment at all, based on that assumption. This sort of thing happens with the breakpoint dose as well. So it is good. So sparse data tends to yield lower point of departure, so that is protective, but there's other areas for the threshold dose, which makes it a bit rubbish. So within the HESI group, we published on what should be done, and we did this in 2013. My old supervisor, Jim Parry, said that scientists like flowcharts. I sort of agree with him. I think scientists do quite like flowcharts. So I came up with this flowchart, and people quite liked it, and it was quite useful. So this is when we were still supporting that no observed effect level and breakpoint dose threshold modeling were quite good. So this could still be used, say, in your analyses if you've been asked to do pairwise testing. This is still quite a useful flowchart for you. It's in this paper. That's me, Johnson, 2013. So that will be in my Google Scholar as well. I think it's free to grab as well. So with this, if we are going to support this approach, a normal distribution, so is it a bell-shaped curve? Just do the Shapiro-Wilks test or something similar. Homogeneous variance, the Bartlett's test. So that means just is every level of variance equal throughout the dose response. If some of them are massive and some of them are small, this really rejects a lot of assumptions from pairwise testing. You'll see in lots of people's publications, they don't do these tests, but they still do the Dunnett's test. That's one indication that they clearly can't do statistics. Most of those findings then become irrelevant and nonsense, but people don't really understand that, so it's quite problematic. If that is the case and these tests are not done or they are just failed, there are ways around this. You can have do the Dunn's test, which is non-parametric version of the Dunnett's test. And if you are going to do these are all available in the standard stats packages like SAS and SPSS and Minitab and MATLAB. I don't know, all the different versions. I think GraphPad's quite a good one that students use nowadays. So you can do all these different ones. So if it's great, do the Dunnett's. If it's not so great, do the Dunn's. And then these are the dose response modeling thereafter. But I think we'll just stop here for today's talk, just to show there are different ways of defining these no observed effect levels. And this, we went into this in some detail. So we did try, but we still think it's a bit rubbish. Threshold modeling. This is quite loaded as well. People use the term threshold a lot. It's still in lots of regulatory guidance. It shouldn't really be in regulatory guidance unless it's threshold mechanism. It's really problematic and really poor science, to be honest, to talk about threshold doses and threshold modeling. And here's a figure from Kenny Crump. So he invented the benchmark dose approach, really. He was on our Brazil IWGT group. And this figure just identifies one of the major issues with it. So if you've got a data set like this, then okay, oh, you applied a threshold model to it. You've applied this broken stick, this hockey stick type approach. So that'd be a American type hockey stick, not probably a European type hockey stick. European hockey stick would be more hormesis, a J-shaped curve, but that's an even more problematic thing that we won't talk about today. So here we got this threshold type model. Okay, there's the threshold dose. There's the lower bound of the threshold dose. Everyone's going to be safe if we do a risk assessment based on that, if this is a good endpoint. But you can actually just apply different models to this data. So you can apply this non-linear model to it. So in this one, there is never a point of departure. So actually, there's no safe dose. This is quite a big problem, but you can just reject that by doing threshold modeling. You could just draw a straight line through this and go like they do with lots of cancer risk assessments and just say, oh, this is linear. Everyone's going to die, that sort of thing. But really, you can see that that's not linear. So it's already prone to quite big issues doing this dose response modeling. So you need to understand what it all means, really. So what this figure really means is that you need an infinite sample size to be able to prove that this model fits these data. So an infinite sample size, so infinite, as many data points as you can possibly imagine and more. Error bars, there wouldn't be error bars, it would just be the diamonds, and then it could fit it perfectly. You've never got an infinite sample size, so you can never do threshold modeling with any precision. So don't. But that's not a problem. We don't need to talk about threshold doses. We don't need to talk about threshold modeling at all. So benchmark dosing overcomes all of this. So what this does, it's been used for cancer risk assessments for years from people like EFSA and EPA. It's really well done. Other areas of toxicology as well, been using this for many years. So it is a good approach, a well-used approach. And all we're saying is, can we do this for genotoxicity data? So here, instead of this, let's force one of these threshold doses to it, or say we know exactly what this data set is. Benchmark dose modeling. So this is how many doses we've got: 1, 2, 3, 4, 5 doses plus control. We've defined what the background is, what we're going to say. So I'll talk about critical effects and size in a bit. So you define what your small but measurable increases above your background. For genotoxicity, this is 50%. So a CES of 50 is what is currently put forward. And then the benchmark dose approach puts forward a series of models against your data and says, oh, it could be over here, could be over here. Okay, let's put a model from there to there, all the way through. Benchmark dose then comes along and goes, okay, worst case scenario is the model could fit here. Best case is it could fit over there. You get your BMD in the middle, or not always in the middle, but you get your BMD somewhere between these two. And then you get your lower bound and your upper bound. And this is your confidence interval. And this is of ultimate importance. If you just remember a couple of things from today's talk, this is probably one of the ones you want to remember. So you've got dose response modeling. Could be there, could be there. Okay, so we're going to agree to that. The benchmark dose lower bound is here. The upper bound is here. And we've got confidence intervals around this. So now we're saying, oh, we've got a point of departure. And we're this confident with that this is where it is. And it tells you a measure of precision as well. You don't have that with the no observed effect level. You don't have that with the TD50 as well from cancer data. So it's got a few benefits even just by having these confidence intervals. And you can do quite a lot with these confidence intervals. So using genotox data for risk assessment to date. So we've been looking at that. We've published these papers, as I said. This flowchart I probably won't go into today. It's probably less relevant for today, just to show that we've been working on this for many years for the IWGT and the HESI group, and we're progressing quite a lot. And now we're at the stage of how can we extrapolate from these animal data to humans, So we're at the risk assessment place. And there's lots of fun things you can do in the middle, with covariate BMD analysis being something I'll talk about in a minute. So if we're not going to use the no observed effect level, the one from pairwise testing, if we're going to use the BMD, how do you do it? So EFSA have come up with this. I think this one's slightly out of date now, but it's still okay for the talk. You can go, this is a benchmark dose flow. So if there's convergence, it tells you whether there's convergence in your assessment. No observed trend. So, okay, if nothing's happening, you can stop analyzing and say no dose response using the tool. No, nested families are selected. This one's with the smallest AIC or model averaging is put forward. This is all done for you. This is done for you as well. This can be rejected, and this gives you an alert if it does overcome the AIC, but you don't have to worry about that too much. All of this is really sorted out if model averaging is available, and model averaging is available, and then I'll show you how to do this in a minute. So there is this risk assessment or BMD advice type flowchart that's given by EFSA 2016, and really just points to, if you've got data, model averaging will be able to sort it out for you, unless you've got some alerts along the way. They've updated it with a new flowchart, which includes Bayesian BMD approach, which I won't talk about. It gets a bit spicy if we talk about that, so I won't talk about that just yet. Okay, so we've got BMD. We're going to show you how to do BMD analysis in a minute. You'll be able to do this on all your data and be able to understand all people's BMD assessments that they've been putting into risk assessments and publications following this. That's my plan. So you do this, apply your statistical models. Critical effect size, remember, is your increase above the background. As a default for continuous data, this is 5% or 10%. But there's a new approach where you can tailor this to your endpoint. And for Genotox, this is 50%. This is not the same as 50% for quantal data, which is, say, cancer data. Quantal data is, say, 1 animal out of 50 has cancer. That means 50% CES for those would mean that 50% of those animals have got cancer. That's not what this is for Genotox, for toxicology. Or a 50% above the background is just like a 1.5-fold above the background. So very different for toxicology compared to cancer bioassay. And that's really important for you to remember. I'm eating into my time, so I will just skip through that one a little bit, but it's good for your reference just to critique why no observed effect level is not very good. But here's a visual interpretation of why it's not so good. So here's that dose response that I showed you before for the BMD modeling. Okay, there's a no observed effect level. You've done your Dunnett's test. That overlaps with that, so it's not significant. That's all great. You got a nice study in this case. You got 5 doses plus control. What happens if you got 3 doses plus control? Okay, your no observed effect level suddenly shifts down there. Is that a true no observed effect level? Is that true safe dose? No, it's not. It's just what doses did you select? So just immediately you can see it's not very good. It depends on what doses you select. The BMD approach is not so affected by what doses you select because it's a modeling approach. So it's immediately better from that respect. Another one is if you just increase that variation for this one to that much, your no observed effect level is shifted from there up to there just by having poorer data. So you're moving your point of departure around just by having poor data or selecting different doses. So to me, it's pretty much meaningless regarding risk assessment. That dose doesn't really mean anything for safety. So why would we do anything with it regarding safety? Basically, we should not do that. And that's what BMD experts have been saying for years. Probably using better words than I've just used, but that's my little ditty for today. We're going to put forward how best you can do study design. So study design does affect how your BMD analysis will come out. So there is some guidance about all of this, and that's what these couple of slides are for. One of the biggest concepts with BMD modeling is, so with pairwise testing, you do your power calculation, and that tells you how good your study is going to be at the start. With BMD modeling, you don't have that luxury. You find out how good it is at the end of your analysis. You can front load it to say, okay, we're more likely to get a good analysis if we do these certain things. And these are those certain things. So the certain things are, instead of, say, 3 doses plus control, you want more doses plus control, say 5 doses plus control. Doesn't actually matter so much how many replicates are within those different ones. Just the spread of data makes it better. So there's a few publications from Wout Slob on this for 3R's benefits of using BMD approach, and that's just basically just highlighted a bit more here. So Ali Long and Mark Beale from Health Canada will publish on this quite soon. So I just thought I'd better highlight that those guys are working on this. We've also been asked to write a parallel monograph, or parallel, what's it called? An OECD, not guideline, but a parallel thing that people can use in line with other gene toxicity test guidelines to do all this as well. So we might write that if we find enough time, but you've seen we're all quite busy already. So that's the plan. We're going to do this dose response modeling now. I think I'll just do it, but I've given you the data through Sabine, I think. So you just go to dose, which is simple, response, which is simple. And this is the bit where I did talk a little bit about the different things. So continuous value for each individual. So that's where you've got the percentages, like the micronucleus. I've done that ramble. So continuous data set like micronucleus or TGR, which is transgenic gene mutation, that kind of thing would be continuous. Continuous summary is micronucleus or TGR or that kind of thing if you've got means plus standard deviation or standard error. And quantile would be, say, cancer data. So the first data I've given you is just a standard continuous data set. So this is the bit where we change the benchmark response or the critical effect size. So it's currently set to 5% or 0.05, so that's 5%. We want to set it to 50%, so that's 0.5. We want model averaging, so we're going to do this. The standard number of bootstraps, so this is just how many times the 4 models are applied to the data, is called bootstrapping. So that's that bit. So this is the most powerful approach, this model averaging approach. If you didn't do this, it would just apply a series of statistical models, and you would then select which model was better based on, say, the Akaki selection criteria. But that's not the best approach. When model averaging, as I've shown from the EFSA approach, when model averaging is available, you should always use model averaging. So that's that. So I think the problem is if I have multiple tabs open, so I won't have multiple tabs open. So now I'll go to data. So I've got multiple data tabs open, so I'm going to close one of them. Hopefully this will work.
Yeah,
so this
is so far so good.
Cool. I'm closing everything and confusing myself, but we're all good. So that data set is in the slide set as well. I'll keep an eye on time too. I'll probably do a covariate analysis as well with you too, or just show you how to do click the buttons and then show you the output. So if you're going to publish using these sorts of data, what you're going to do is show what you did. You used a 50% critical effect size. You did model averaging, number of bootstraps. So these will all be important to put into your publication. This is the overall output. So what the modeling does, it applies these 4 statistical models, so inverse exponential, this LM model, exponential, and Hill model to the data. You can see they all fit the data quite nicely. So that's good. The outputs are on the right, and these are just the 4. This statistical model has been applied to this data. These are the BMD metrics. That's the BMDL, that's the BMD upper. CD, so that's the BMD midpoint. And these are other parts, other parameters that are specified as well, or calculated. We look at all the different fitted models, and these numbers we don't have to worry about too much. You did have to worry if we weren't using model averaging. You would use this Akaki selection, this AIC number personally, but you don't have to with model averaging. And then what it does, it weights the models to a greater extent. So this inverse exponential isn't working very well. So that's fit the data to a poor degree. It hasn't got this little curve at the top. So it's not weighted so highly. So the BMD modeling will be more weighted with this within the bootstrapping. So you end up with this 200 bootstraps, more weighted towards these other 3 models, and then you get your BMD metrics out of this. So in a publication or report, you put all of these in, you put that in, and then your metrics are down the bottom. And this is your model weighting. So it's pretty easy. The hard bit is getting these decision points right. So that's why we're trying to provide guidance to everyone. It can go wrong, and there can be times when you want to do something else. One of the times when you want to do something else is if you want covariate data. So I've given you a covariate data set. I think I will go through it. I'll risk it. I think it's worth it. I think showing you how to do this is probably more important than me rambling on. So here's a covariate data set. let's call it COV, I'll call it SAB. So the data set is like this. So here we've got 3 doses. So that's 1 replicate, 2 replicates, 3 replicates for dose 0. These are study. So this might be a compound. You might be testing an antigen to another antigen. And then we've got 3 of those studies. So here's one. So this is how it needs to be laid out. You need these 3 columns. You don't really want spaces. So the difference between that, I've just put a dot, a full stop. And then you end up with these 3 columns. So you've got dose, response, and what study it is. So when you put this forward and you do the analysis on it, you got dose, you got response. This is continuous data again. It's not summary data. It is covariate this time. So this is different to last time. So that'd be study. Still do 50%, still do model averaging. And this will take ages because it's quite intensive on the statistical models. The covariate analysis and the model averaging will take bloody ages. So you wouldn't be able to do this on an old-fashioned computer in the old days, but we can nowadays. I'm just using, so I think this is just on their internal server at RIVM. It's not even a supercomputer or anything. So with this, what you get out of this is quite nice. You'll see some of my publications will look like this. I'll put these in. So these are, this is the model. That model's fit these data quite well. These are the different studies. So the green one is study 3 over there. The red one will be study 2. And the black one will be study 1. So you can just see, if you're going to eyeball these data, you would say, this compound's more potent than that compound. These compounds are quite similar in potency, but now we can say, okay, compound 2 is more potent than compound 1. So we immediately get quite a lot of information out of this. Does the same sort of thing, still model weighting. You can see the exponential model has fit the data set to a much greater extent. So the exponential model here, it's got that little kink at the top, a little plateau. That's obviously what the data are telling us. So it's fit the data to a greater extent. Then you end up with these 3 sets of BMD coming out of this. And this nice output called a potency ranking. So you can say this compound's, like I said, not so potent. If they're overlapping confidence intervals, you can say, okay, they're equipotent. So it's really good. Another good thing about this as well is this improves the statistical analysis. So potentially, if you just modeled this data set 3 by itself, you might get quite poor BMD confidence intervals. So you might get quite poor, quite wide confidence intervals. But this approach models all the data at the same time, and you get information from this study impacting on the model fit of this study. So you get improved higher precision by carrying out this approach. We published 2 papers on this in 2016 by John Wills et al. To show all of this. And at the end of my talk, there's loads of examples of this. So I really like covariate analysis. I'll show you just one last one. So it's with quantal data, same sort of thing. If you're going to study cancer data, you just go into the GOLD database, the LASA database now. You put it in, and then you get the confidence intervals out. Again, model averaging just does all the tough work for you. So that's that. Within this slide set, those are the data sets. These are the outputs that I've been getting from those analyses. You should be able to do it, so you can see, okay, can I do this or not, based on those data sets, and we go from there. So we're going to publish on why we use that critical effect size of 50%. Just make sure I'm all right for time, so Sabine doesn't have to tell me off. Need to slow down. Okay, I've got a bit more time if I need it. So we've got this IWGT. So that's the International Workshop on Genetic Toxicology. We met up in Ottawa, so that was another nice conference. I think there's a nice pub crawl at that one. Nice hotels, good science, and it was really good. And within that, we did agree that 50% was the critical effect size that should be used for in vivo gene tox data. We are publishing that this year. These papers take quite a lot of time. We've got people from regulatory bodies and industry people sort of arguing about different aspects, and people are busy, so these papers take a bit of time. But basically, you could then cite that paper and say, okay, 50% is what this expert group says to you, so I'm going to use it. Published a few other ones as well. This is in vitro to in vivo extrapolation. Some of you in the room will be going, oh, I don't really care about this in vivo work that he's been presenting. So this type of work is coming out as well. So in vitro to in vivo extrapolation, we do the same sort of thing with the in vitro data. You do benchmark dose analysis on all of this. And then you can create human equivalent doses through just some quite simple mathematical modeling around dose. And those are all explained in these publications, and those have been part of HESI and IWGT2. So I think the in vitro risk assessment side of things is progressing very well as well. I have a lot of time for it. At a recent expert group in BFR in Germany, they really were quite supportive of quite a lot of the in vitro approaches. That PARC big EU project is pushing forward with EU approaches and quantitative AOPs and stuff as well. So I really think there's quite a lot of mileage in this sort of approach and parallel approaches using in vitro data. Not sure it's quite there yet, but do try these things out and do publish on this area. And I think this is going to progress quite quickly. Obviously, dose is a bit of an issue, metabolism, but lots of those things can be extrapolated and modeled quite precisely nowadays with these quite decent statistical packages and modeling tools. So I mentioned that I do quite like this covariate approach. There's lots of different things you can do with the covariate approach. I've shown you how to do it online. If you're going to do it on your computer at home, you can do this on bigger data sets too. Many people at home have got quite good PCs, so I'm able to do like 20 compounds at the same time. I think when I started out, that would take 2 days to analyze, and now it takes about 5 hours to analyze. So things are getting a bit better from computing side as well. So with that, if you have these 3 data sets, compound A, B, and C, you just eyeball them and go, okay, compound A is more potent than compound C. What's the metric? Is it going to be the no observed effect level? No, no observed effect levels are rubbish. They are not a measure of potency. They should never be used for potency comparisons. Okay, let's not do that. Let's do this. So BMD covariate analysis, this one doesn't overlap with that one, doesn't overlap with that one. So compound A is more potent than compound C, and compound B is in the middle. And that's it. Quite easy to do, quite easy to generate these hypotheses. And you've got a convincing figure at the end of it, then cite these papers and say, this is how you do potency ranking, and these are my findings. We like to push things quite hard, so we did a different thing as well. So this is for one endpoint. This is for 2 endpoints. So you do it for endpoint 1 down there, endpoint 2 up there. So that is that compound, that one is that compound for this endpoint. You then plot that against what it would look like for the other endpoint. And then you go, okay, for this compound 1, it's more potent in this assay. It's also more potent in that assay. That means that that assay links to that assay. Obviously, that opens up quite a lot of opportunities for saying, this is how you could validate one test against another test. So we did that with Tox Tracker, with Heal in Toxis. I've also done it for different groups as well. We did it for micronucleus endpoint compared to cancer. We're now doing it for in vivo mutation to cancer. And it's a really good way of comparing 2 different endpoints. And it has been very, very useful. The next step is then to just bypass the need for that other endpoint and do risk assessments based on one endpoint. And that has been done once in RIVM, and it's been put forward by others as well. And it's also an approach that might help us out of this nitrosamine issue with banning 40% of all drugs from the market based on nitrosamine impurities too. So it's getting there. It's useful and it's good. A few other case examples, just so you got some things to cite and read about, would be like this one. So these are different antigens. I talked about antigens previously. You got flubendazole, oxybendazole, mebendazole, albendazole, benamyl and carbendazim, albendazole oxide. So these are all quite similar antigens. They act at different positions on the microtubule. There's different binding positions on the microtubule. So they do slightly different things, or they do quite similar things. So benamyl and carbendazim are structural similarity. They're structural analogues of each other. One's a metabolite of the other. I think they're pesticides, I think. These ones down here, these are anti-parasitic compounds. So you find that quite a lot. If something's good at breaking the microtubule, it's also good at killing off parasites that work by swimming by or dividing by the microtubule as well. So those compounds are quite equipotent. Flubendazole and oxybendazole, equipotent. Albendazole, not as potent. The oxide version of albendazole is much less potent, and these ones are different to these ones. These ones are similar to each other. immediately, just by doing these covariate plots, you can interpret your data a lot quicker, a lot more precise, than just having a big data table. So it's been quite beneficial. Loads of other things you can do with it as well. We've been linking it to topoisomerase inhibitors. We've been validating test systems with it to say the flow site, the imaging flow cytometry version, the micronucleus assay, is as precise as the manual version using this sort of approach as well. They both come out with the same sort of metrics. So it's been quite good along the way. A different tissue is more sensitive in an endpoint. Yes, they are. Sometimes they're more sensitive. Can you do this based on no observed effect level comparisons or TD50 comparisons? Sort of, but not very precisely. The best way is to do benchmark dose analysis and say, okay, over here, benzoate pyrene, small intestine, is more sensitive than these up here, and so on. So we get lots of information out quite quickly. I think that's probably about where I should stop. I think I just one more taster. Sorry to say that. I hate false horizons. People sighing to say, "Oh God, he's going to shut up now," and he's not. So this one last bit, definitely one last bit, is on ToxPy. So we're looking at how are these the best way to present these data? Potentially not. For some of these multiplex assays like the multiflow or tox tracker, you might have multiple data sets. You might want to do something in a more visual way, and you can plot them all in these ToxPy plots. You end up with potency scores with all of these endpoints together. So you can see H2AX is there, H3 is in there, P53 is in there. Mitomycin's more potent than all these other ones. You have these potency ranks for these multiple endpoint schemes. You can do these cool things where they start grouping together based on hierarchical clustering, and that gives you information about different things as well. We just published this paper, 2024, on all of this, and showed that these non-genotoxicants all grouped up together based on the BMD of these multiplex endpoints and cytoclastogens and so on. So have a little read of that paper, and I promise that is the end. And these are the people I thank, and hopefully we can get to questions without loads of weird noises and me sharing something weird. So I will stop sharing and look forward to any questions. And again, I can be followed up with by email, and at the bottom of this is my email address and my LinkedIn as well. So please do contact me if you want more information. Thank you.